everyone, welcome to Learner's Biology channel. Myself Sakira Banu. I am an MSc Zoology from Guwahati University and I'll be dealing with your biology lessons. So today we are going to start the first chapter of biology, that is chapter number 5, the fundamental unit of life. So I hope all of you know what is the fundamental unit of life. Obviously, it is the cell. So without cells, no living organisms can be formed. So firstly, we will move to the history of the cells. So while examining a thin slice of cork, Robert Hooke, he saw that the cork resembled the structure of a honeycomb which consists of little compartments. So this person, Robert Hooke, in the year 1665, he found out that the cork of a tree, cork is a substance which comes from the bark of a tree. So this cork, it consists of little compartments. So in the year 1665, Hooke was able to see this observation through a self-designed microscope. So since the cork consists of little compartments, Robert Hooke named these compartments or boxes as cells. So cell, it is a Latin word which means a little room. Now this discovery by Robert Hooke, it may seem to be a very small and insignificant incident, but this discovery, it was very important for the history of science, because before Robert Hooke, no person knew that the living things are made up of such kind of compartments. So this was the very first time that someone had observed that living things appear to consist of separate units. So Robert Hooke, he was the first person to find out that living things consist of some separate units, and these units he named as cells. Now we will find out more about cells. So what are living organisms made up of? For this, we have to come to an activity. So what we have to do is, we will take a small piece from an onion bulb, and with the help of a pair of forceps, we will peel off the skin of the onion bulb. The skin of the onion bulb is known as the epidermis. So from the concave side or the inner side of the onion, we are going to peel off the skin. Now this layer, it must be put immediately in a watch glass which contains water. Doing so, we can prevent the peel from getting folded or getting dried up. Next, what we do, we take a glass slide, and on the glass slide, we'll put a drop of water, and then we will transfer the peel from the watch glass to the slide. We have to make sure that the peel is perfectly flat on the slide. So for this, we can use a hair paint brush to transfer the peel into the slide. Now to view the cells, we have to put a dye. Okay, so what we do is, we will put a drop of safranin solution. So safranin is a dye which gives color to the cells. So what we do is, we will put a drop of safranin solution on the piece followed by a cover slip. So safranin solution is a dye which will color the onion peel, and later on we will put a cover slip. While putting a cover slip, we should be very careful to avoid air bubbles. Okay, so for this we will take the help of a mounting needle. So this mounting needle will help to avoid air bubbles and put the cover slip properly. In this way, we can prepare a temporary mount of the onion peel, and then we can observe this slide under low power of a compound microscope. Now this you can see is the compound microscope. So this compound microscope, it has different magnifications. So as you can see here, this is the base of the compound microscope, and here lies the mirror. This is the stage. Okay, so on this stage you can put the slide. Then these are the objective lenses of different magnifications. Then this is the arm which you can hold. Here you can do the adjustment. This is the body tube, and here lies the eyepiece where you can put your eye and watch the cells or see the cells of the slide. When you observe the onion peel through the lens, you will see a structure of this sort. So these are the cells of an onion peel. So here you can see small compartments, right? So these small compartments, they are known as cells. In the middle of these cells, you can see a few dots. Now these dots are the nuclei of the cells. So we can try preparing different mounds of peels of onions of different sizes. So we can collect onion peels of different sizes and can prepare temporary mounds. Now, what are these structures? These structures look similar to each other and they together form a big structure like the onion bulb. That means the onion bulb is made up of a number of cells. So from this activity, we can find out that onion bulbs of different sizes, they have similar small structures. So regardless of the size of the onion, whether you take a big onion peel or a small onion peel, that doesn't matter. The structure will always remain the same. So these small structures that we can see under the microscope, these are the basic building blocks or units of the onion bulb. And these building units or structures, they are named as cells. So not only onions, all the living organisms that we observe around us, they are made up of cells. But here, one point to be noted is some organisms are made up of single cells and they can live on their own, while others are made up of a number of cells combined together. So here we can know more about cells. So as we have already learned that in the year 1665, Robert Hooke, he first discovered the cells. Then in the year 1674, another scientist or another person named as Leeuwenhoek discovered the free living cells in the pond water for the first time. Now the pond water, it has different free living cells, zooplanktons, phytoplanktons. So these were discovered by Leeuwenhoek in 1674. In the year 1831, Robert Brown was the first person to discover the nucleus in the cell. So before that, no one knew that the cells contain nuclei also. In 1839, Purkinje, he coined the term protoplasm. What is protoplasm? It is the fluid substance of the cell. So the cell contains a fluid substance, a liquid-like substance, which is known as protoplasm. The next discovery was done by Sladen and Schwann in 1838 and 1839 respectively. So Sladen and Schwann, they proposed the cell theory. Now what is cell theory? The cell theory says that all the plants and animals are composed of cells and the cell is the basic unit of life. So this is the cell theory which was proposed by two biologists, Sladen and Schwann. The cell theory, it was further expanded by Virchow in 1855, where he suggested that all cells arise from pre-existing cells. That means he suggested cell division. A pre-existing cell divides to form a number of cells. And in the year 1940, with the discovery of the electron microscope, all the complex structures and the organelles present in the cell were visible. The invention of magnifying glasses led to the discovery of the microscopic world. So, before that, no one could view the microscopic world. But Only after the invention of the magnifying lenses, invention of the microscopes, one can view the microscopic world. So it is now known that a single cell, it may constitute a whole organism, as in amoeba. Then you have chlamydomonas, paramecium and bacteria. Now these organisms, amoeba, chlamydomonas, paramecium, bacteria, these are all made up of single cells and they are known as unicellular organisms. On the other hand, many cells group together in a single body and assume different functions in it to form various body parts in multicellular organisms, such as some fungi, plants and animals. So, some fungi, plants and animals, they are made up of many cells which are grouped together. And these cells, they assume different functions to form various body parts. So, these organisms, they are known as multicellular organisms. Now, every multicellular organism has come from a single cell. Now the question arises, how? Here lies the answer. This is because of the cell division. So all cells, they come from the pre-existing cells. So the pre-existing cells, they divide and divide to form the new cells.
So students, we end our class here. In the next class, we'll be discussing the rest of the portion of this chapter. Thank you everyone.